Our project is on bacteria versus plastic. The bacteria that we used is Bacillus subtilis, and the plastic that we used is known as microplastics. But the microplastics that you may have heard of are probably macro that you can actually see with your eye. The microplastics that we used in our lab were things that you could only see underneath a microscope. And so our original purpose is to ultimately rid microplastics from all water mediums to protect all organic life, aquatic and humans alike. For our question, we wanted to see how effective Bacillus subtilis is in removing microplastics from all waters. And our hypothesis is that when we add Bacillus subtilis into the water mediums, it will in fact remove and degrade the plastic, therefore helping out our ecosystems and improving all organic life.
And you may not know it, but there are microplastics all over your entire body and every organ that you have. And they inhibit hormone production, and they hurt your cognition and things like that, and reproductive development as well. But in order to do our project, we needed to get this bacteria. So we ordered it from a company based out of Europe, and we cultured it on 6 different petri dishes. And it grew over the next 3 weeks after we cultured it. Then we took off 1 gram worth of this Bacillus subtilis bacteria, and we put it into 4 petri dishes, which we have pictures of here, that were made of the microplastic contaminated water and a nutrient broth. We also tested different salinities. So over here is fresh water. Up here is clean drinking water, but we found actually does still have microplastics, and this is just a reference photo. This one, you can see the colonies of bacteria. Each of these little dots is a colony of bacteria. And this is a much more zoomed in version of this. And as you keep going, the salinity becomes increasingly larger. And down here is mimicking ocean water. And then for some of our variables, our independent variable was salt, which is because we changed it. Our dependent variable was the efficiency of Bacillus subtilis to degrade the microplastics. And then our control variables were like the microplastic water, the nutrient broth, the microplastics themselves, and things like that.
So over here on the right side of the board, this is the data that we collected. Initially, this is a 0.01 gram sample, and we counted 34 microplastics right here. Scaling it up, all of our samples with bacteria in them had 25 grams of this water each. So we roughly estimated 60 to 80,000 microplastics per sample. Now, when we put the bacteria in, we were trying to measure the growth, and noticing here on the bottom where there's 0 salt at all, the fresh water, we can see very, very large clumps or colonies of bacteria. And comparing this to the half of the ocean water salinity, you see there are colonies, but they are much smaller. And then 3/4, they're even smaller. And in the full ocean water salinity, there's almost no colonies at all. This behavior is very irregular. And when the bacteria is alive, it will reproduce and grow and form colonies. So we thought it was safe to assume that for higher salt water, the bacteria is dead. And looking at our data, as we said, 60 to 80,000 microplastics per sample. Each one of these has 1 gram of the Bacillus subtilis bacteria. And so we calculated over the 2-week span, each of these was left for 2 weeks. We estimated roughly 2,400 to 3,400 microplastics were being degraded per milliliter of water. And how that process happens, we detail a little bit here. Since the plastic is a synthetic material, there's no bacteria that will actually just eat the plastic. Instead, it eats the nutrients in the water and excretes multiple enzymes that end up breaking down the plastic. One of them was anethase right here. This specific enzyme connects to the starch in the plastic and breaks up the chains. It leaves behind some sucrose in the water, so that is a side effect. But of course, this is not as bad as the plastic is, not as dangerous for your health. And that's just one of the enzymes that excretes. There's multiple here, such as lipases, cellulases, beta-glucans. There's many different enzymes that each target one specific part of the plastic to help break it down. And that's what we noticed.
To carry on further, we have ideas for the future of this project. We have seen that it is capable of breaking down plastics in fresh water, which would mean that it is able to be used in polluted freshwater areas like lakes, rivers, and things like that. Additionally, as we did say, it dies in salt water. But we have resources, and we might be able to find even more resources to be able to genetically modify our bacteria, which would be very beneficial for our project. Being able to allow this bacteria to survive in a ocean-style water with a lot of salt in it, which we've seen it dies here, would start saving the world, because these plastics are in everything and they're found in every body of water. They're all over the ocean, and right now we're being stopped at fresh water. But being able to take it to the ocean would be a great step for our project.
And with our project, what we really hope to do with all this research is just cut it off where it begins, because microplastics, they can come off of clothes, they come off of water, and that's why we started studying water. But honestly, the microplastics come off of anything plastic. The food that you microwave, it's just everywhere. But if we can cut it off at the water that cows drink for meat, cut it off at the fish, cut off at the water that the fish would be like swimming in and consuming their food in, just all water in general, we would limit the microplastics that are in our world and help every single organic living thing in our world. And I think for the future, like what my brother said, we're going to try and bioengineer our Bacillus subtilis to be able to live in harsher climates where the salinity is about the average of ocean, where we did find it didn't survive, or also at varying temperatures, because this experiment was conducted at a very set temperature of 37 degrees Celsius.
I would like to add one final thing about the bacteria itself. It is commonly found in fresh water, it is commonly found in soil, and it is also a probiotic. So at this very moment, it is within every living animal's intestines. It breaks down your food for you, but it can also break down plastic. So it is completely safe for you to consume.